Welcome everyone, welcome back. I think, well, for me personally, yesterday was a very good start to IMUC-III. I found it scientifically, intellectually stimulating, but also emotionally quite stimulating. We heard from some of the doyens of Buruli ulcer over the last 4 or 5 decades, and that's a special thing, especially for those of us that have worked with these people. But today, this morning, we're going to switch gears and talk about the interaction of the pathogen with the host, and in particular, the role of mycolactone in Buruli ulcer and beyond. But we'll come to that in a minute when I introduce one of our chairs for this session. In your tote bag that we put together for the conference, you'll notice a pair of socks. And you may have noticed that there's a watermelon pattern on the socks. It's not a watermelon. It's Arvind Yaramilli's careful mapping of where Buruli ulcers present for patients here in southeastern Australia. When you wear these socks, and we hope that you do frequently, and people say, oh, they're beautiful watermelons, no, you can explain that's not watermelon, that's Buruli ulcer, and you can use that as a prompt to describe the work that you do, why it's important, and what you're trying to do to eliminate Buruli ulcer. So wear the socks with pride. Yes, they can be watermelons if you like, but try and use them as a tool to promote the work that you do around Buruli ulcer. Okay, now we have got we've got a wonderful day lined up today because at the end of our scientific session, we're going to eat our lunch and then go into a bus and head down into Buruli country. Buruli country is dangerous because you can get Buruli ulcer, but it's also very beautiful. So it'll be a mixture of fear and wonder. The fear should be very, very small because the chance of you getting a Buruli ulcer is tiny, but the wonder should be high because it's a beautiful day and it's a very nice part of the world. Paul Johnson.
I was wondering whether the socks are guaranteed to prevent Buruli ulcer.
No. What we've found, the proboscis of our mosquitoes will penetrate through the material of the sock, and
directly onto the black dots.
Right, yeah, that's right. Yeah, the socks are a targeting system. We didn't think about that. All right. Well, I'm very pleased to introduce one of our co-chairs for this session, Professor Heather Jordan from Mississippi State. And Heather, over to you. Thank you.
Thank you so much. It's a pleasure to be co-chair to this section where it's also a pleasure to introduce one of our keynotes. Hey!
Sorry.
It's my pleasure to introduce Steve, the co-chair. So it's a pleasure for us to introduce our first keynote speaker, who is Dr. Rachel Simmons, who is a wonderful friend, colleague, partner in crime in many ways. But Rachel is a professor of immunopathogenesis, head of the Department of Microbial Sciences at the University of Surrey, and chair of the WHO Skin NTD's sub-working group Basic Research. Her Buruli ulcer research group primarily focuses on the mechanism of action of mycolactone, and in 2014 identified its mechanism of action as an inhibitor of the Sec61 translocon, as well as continuing to advance our understanding of how this molecular mechanism drives Buruli ulcer pathogenesis. The group is also involved in interdisciplinary research for BU and basic cell and molecular biology of Sec61 inhibition by mycolactone and other compounds. So, my pleasure to introduce Rachel. Thank you, Rachel. All right. It's also a pleasure to introduce our second keynote speaker, Professor Caroline Demangel from the Institut Pasteur, where Dr. Demangel studied life sciences at the National Institute of Agronomy at Paris-Grignon, where she completed her PhD in cell biotechnology. Since 2011, she's been directing a research unit in the immunology department, where she currently serves as deputy director. Her group investigates the microbe-immune system dialogue to identify novel bioactive molecules and therapeutic strategies to manipulate immunity. Caroline, you have the floor. Thank you.
Thank you for the introduction, and thank you also for the invitation. It's a real pleasure to be here and to see you all again after all this time. It is a wonderful community, so I'm very glad I'm here today with you. So the title of my talk is a bit different from what you saw in the program. I will focus my talk around the immunosuppressive properties of mycolactone, and this was agreed with Rachel so that you have a quite comprehensive view of all the biological properties of mycolactone. So for me, this research started 20 years ago in the unit of Stuart Cole at Institut Pasteur. I was coming back from my postdoc in Sydney, and I was very lucky to meet in Institut Pasteur Tim Stinnea doing his postdoc there, and Paul Johnson doing a sabbatical. And I discovered through them this other mycobacterial disease. I was essentially working on TB at the time. And these two guys did not transmit me BU, but they transmitted me their passion for BU. And this is very contagious, obviously. And I've been working essentially on mycolactone since then. So I'm not going to introduce to you the different stages of BU development. This is just a slide to introduce the reasons why I got very interested in this disease. As an immunologist, I found extremely intriguing the relative lack of inflammation and pain that characterize BU lesions. So it's not black and white, right? It's dynamic too, but the fact that these lesions are poorly inflamed and reported as painless is very surprising considering the amount of bacteria and of dead cells in these lesions. So this suggested immunosuppressive properties of the mycobacteria, and the fact that these defects resolve when the bacteria are eliminated by surgery or antibiotic treatment very much supported the hypothesis that bacteria do something that suppresses the immune system. So it was noticed very early on that there was this lack of inflammation locally, and also that there were defects at the systemic level in T cell capacity to produce cytokines in particular. So a lot was already introduced, so I'm going to go quickly on the introduction, but just to reiterate the fact that pioneering work from Pam Small and co-workers and others showed that mycolactone plays a really key role in all the manifestations of the disease. Mycolactone explains the toxic effects on cells. This was shown in vitro and in vivo, and Rachel explained that very nicely. Other manifestation, vascular dysfunction as well, and mycolactone also explains the lack of inflammation locally and the defective responses at the systemic level. So just to detail a little bit further what I mean by immunosuppression, beautiful work performed by the team of Gerd Plüschke and analyzing the histopathological features of lesions showed that indeed, when you look at the tissue, it's not a total lack of inflammation, but you would expect much more inflammatory infiltrates in these lesions that are full of bacteria and dead material. And also, in collaboration with Richard Phillips, we noticed that in the serum of these patients, the level of proteins, circulating proteins that are playing a role in inflammation, it was decreased and as soon as the nodular stage of the disease. And most importantly, when T cells from these patients were purified and stimulated ex vivo with mitogens, they had a defective property, a defective ability to produce cytokines, and very importantly, this is in conditions where the viability of the cells was not altered. So mycolactone is indeed immunosuppressive, and we now know that this is mediated by Sec61 blockade. So maybe a new introduction or another introduction of Sec61 function here to really convince you that this is a key pathogenic mechanism in BU pathology and evolution. So mycolactone is a diffusible product that will enter into the eukaryotic cells, the patient cells, and will localize in the channel Sec61, which is the entry gate of the secretory pathway. And by doing so, all the proteins destined to be translocated in the ER to be either secreted or anchored in membranes will be destroyed in the cytosol by the proteasome. So a very general mechanism, not only affecting immune cells, affecting all cells, but affecting a lot the functions of immune cells. And this is what I'm going to detail a bit further now. So what we showed was that mycolactone is able to operate by direct binding to Sec61, and it was not the first inhibitor of Sec61 that was described. It was the second, the first one being cotransin. Cotransin is another natural product made by fungi, and cotransin, unlike mycolactone, is a partial inhibitor of Sec61. So it leaves the translocation of certain clients normal. Unlike cotransin, mycolactone blocks absolutely everything. And there were cryo-EM studies, recent, some by Rachel and collaborators, and others comparing mycolactone with other natural products that show that mycolactone is binding Sec61 in a different manner. There are common features with other natural inhibitors, but also distinctive features, making mycolactone able to seal completely the Sec61 channel and preventing the translocation of anything. So it was the first super potent total inhibitor of Sec61. So Sec61 has been introduced previously. I just want to repeat here that Sec61 is important for the biogenesis of a number of proteins. All proteins, including a signal peptide or transmembrane domains, will be, during translation, be recognized by Sec61 to enter the ER and proceed their way. So cytokines are perfect Sec61 clients. They will be deeply affected by Sec61 blockades. Cytokine receptors as well. So this is really important. And to pursue a little bit the question on IL-1 beta. IL-1 beta is special in the way that it is not a Sec61 client. It has a different secretory pathway. However, the receptor for IL-1 beta is a perfect Sec61 client. So without receptor, IL-1 beta won't be able to signal anything. However, as soon as IL-1 receptor beta production will be restored when mycolactone is washed away from the organism, it could indeed trigger very potent signaling because there will be a lot of cytokine around in the tissue if it is persistent enough. Okay, this is for the molecular mechanism at the cellular level and on single proteins. But what is going on at the proteome scale? We were interested to study that in immune cells, and we conducted a series of proteomic studies in T cells, dendritic cells, also neurons, to see what is the consequence of Sec61 blockade in different cell types. And here I'm showing you in a very succinct way the results for T cells. In this experiment, T cells were activated by PMA ionomycin, and we compared the proteome in the presence or absence of mycolactone. So on this bar represents the total proteins that we could identify, so around 5,000, and very typically there's about 10% of these proteins that are Sec61 substrates. When the cells were treated with mycolactone, we noticed in the condition of this assay 50 proteins were downregulated, and not surprisingly, many of them in orange here were Sec61 substrates, so very consistent with the mechanism of action. Less obvious, we had also proteins downregulated that were not Sec61 substrates, and this was surprising. These proteins were cytosolic, nuclear, but they shared the property to be all induced by interferon gamma. So what we think is happening is the following mechanism. In a T cell that is activated, what is normally going on is that there is interferon gamma produced, there is interferon gamma receptor expressed, and there is an autocrine signaling loop going on, ending with the transcriptional activation of a number of genes, ISGs, but this is occurring at the gene level. In the presence of mycolactone, interferon gamma production is prevented. It's shown here. Interferon gamma receptor expression is inhibited. So this autocrine stimulation loop cannot happen, and therefore the induction of ISGs is also prevented. So this is just to illustrate the fact that also the molecular mechanism is very clearly Sec61 blockade. At the cellular level, very rapidly, there will be a number of secondary effects on non-Sec61 clients, and therefore very complex protein signature in cells exposed to mycolactone, and very much cell type specific. All right? So why is that important for the Mycobacterium? Well, the interferon gamma receptor signaling axis is key in immunity to mycobacteria in general. This is true for TB, this is true for BU as well. In the presence of mycolactone, T cells no longer produce interferon gamma, macrophages no longer produce a receptor, and therefore the induction, transcriptional induction of this enzyme, for instance, iNOS, that is responsible for the production of reactive oxygen species that are toxic to bacteria, and mycobacteria are very susceptible to them, is prevented. We showed that this signaling axis is very efficiently prevented by mycolactone. So in conclusion, by blocking Sec61, mycolactone is paralyzing the immune system and preventing the generation of protective immune response. It's a very efficient immune evasion mechanism. This is not the only one. I've illustrated today the effects of mycolactone on cytokine production and signaling, on the bactericidal functions of macrophages, but many other functions of immune cells are also affected. Their migratory properties, because it's also mediated by membrane receptors that are susceptible to Sec61 blockade. The antigen presentation by dendritic cells is also deeply affected. So many functions of immune cells are affected. But again, immune cells are not the only cells affected by a Sec61 blockade. They're just functionally very susceptible to this mechanism. How do I do with time?
Oh, you've got 3 more minutes.
3 more minutes. So I'll skip this, because I'd like to spend a few minutes on this assay. So as Rachel was stating, there's the very important question of how much mycolactone circulates in the body of patients, in the tissues, in the blood, and many of us have struggled trying to find good protocols, good methods to do that accurately and with high sensitivity. It's difficult. The lipid extraction and mass spec analysis proved to be not very good in terms of rate of recovery of the toxin. So what we're trying to do is to use the ability of mycolactone to block Sec61 to design a cellular assay. So this is not a field assay, this is a lab assay, but it should allow to assess the concentration of mycolactone quite accurately and high sensitivity in biological samples. So the assay is super simple. We have modified a cell line to express a Sec61 client protein, very susceptible to its blockade, IL-6. And this is a fusion between the signal peptide of IL-6 and the luciferase. So the cells that are transformed produce a lot of this fusion protein that is released in the supernatant, and this can be assessed by luminescence measurement automatically in multi-well microtiter plates. When these cells are exposed to mycolactone, there is a nice dose-dependent inhibition of the secretion of this fusion protein in the supernatant. This assay was performed with human sera spiked with purified mycolactone, and we can detect very low concentration of mycolactone. So in theory, it should allow to measure nanomolar concentrations of mycolactone in human sera, and this is what is going to be tested now in the group of Richard Phillips to address important questions, effect of treatment on circulating mycolactone, correlation between that and paradoxical reactions, and other questions. And in theory, it could be applicable to other types of biological samples. It will just have to be optimized and have good controls. So more in the next IMUC. And that's all I wanted to say today. I would like to thank my group. The work I showed you was mostly done by Laure Guenamassé, who is still in the lab. And many of you have met her previously. And I'd like to thank also my collaborators and sponsors. Thank you very much.
Any questions for Kevin? We have time for maybe 2 questions.
Were you live? Yes. So I was wondering, are there host cell proteins that regulate the expression of Sec61 as well?
Yeah, well, Sec61 is essential. Some cells produce a lot more. For instance, plasma cells who make antibodies, they're really full of endoplasmic reticulum and Sec61, and are very susceptible to mycolactone toxicity because of the effects that Rachel described. Now, the regulation of Sec61, it's regulated to some level in response to stress, but it cannot really be, expelled.
Any possibility that that is different between susceptible and resistant species?
Between?
Say like the difference between cats and possums.
Possible. It's already very variable inside the human organism between cells and tissue, so yeah.
Caroline, could you speculate on the effect that circulating mycolactone might have on things such as long-lived memory cells? So could there be some kind of long-term effect after the Buruli ulcer is treated because of the damage to immune cells, such as a circulating memory cell?
Well, the binding of mycolactone is non-covalent. So I'd say, I don't know if you agree, Rachel, that as mycolactone is, production is stopped by treatments, mycolactone will be with time eliminated totally and should not after a while, but this need really assessment, precise assessments. It should, be completely eliminated. So I don't think that can happen, but to be tested. Yeah.
Good morning. Thanks for a great presentation. Very elegant work. This isn't a question, but we're completing a drug trial among patients with Buruli, and we've got serum samples for pharmacokinetics. We may be able to share them with you to look at your bioassay that you described.
Well,
if that helps, I think that'd be really interesting.
Collaboration starting right now. All right, well, let's thank Caroline. Okay, our next speaker is my fantastic co-chair, Dr. Steve Muhi, who is an infectious disease physician at the Royal Melbourne Hospital with an appointment in travel and tropical medicine clinic, which now sees more patients with Buruli ulcer than ever. He is also a graduate researcher, almost finished, I understand, and is going to talk about some of his work today. Thanks, Steve.
Thanks so much. All right. So thanks very much for the kind introduction. Thank you very much for the invitation. Tim told me that I had to present at this conference, so here I am, but squishing it into a 10-minute slot, and I'm going to try to keep it even shorter than that because we've got morning tea coming up. So I just want to start off by thanking, in fact, some people in the audience who've really helped me along the way through this project, particularly Tim Stinney, who's my PhD supervisor, but there's some others in the audience, Paul Johnson, Dan O'Brien, some others in the lab, Jess, Abel. It's some fantastic collaborators that have helped me along the way. So this is a fairly ambitious project that I nearly completed as part of my PhD here at the University of Melbourne. I am an infectious diseases specialist, so I've got a special interest in Buruli ulcer. I see a lot of it at the moment. So I'm going to talk about a controlled human infection model for Buruli ulcer. So one thing that I'm really nervous about is, and I was talking to some colleagues yesterday, is about the increasing number of cases of Buruli ulcer, particularly in Melbourne, but all across Victoria. And I'm interested to know where we're going to be in 20 years' time, because at the moment we're struggling actually. So the reason why I was late was because we've got so many patients in this trial, in this Telesibec trial. We have so many patients to recruit for novel antibiotic strategies that we're actually struggling to keep up to a certain degree and filling recruitment very quickly. So where are we going to be in 20 years' time in terms of our epidemiology? This is really driving me to think about innovative approaches to try to tackle the re-emerging threat of Buruli ulcer, and particularly in Victoria. Trying to think about ambitious ways that we can identify models to better test preventative and therapeutic strategies, but also to better understand the immunobiology of the disease process itself. This is a very controversial topic. I think Paul Flood introduced it yesterday very briefly in the Q&A session. But this is a topic that's got a lot of stigma associated, as mentioned a couple of days ago, or yesterday morning, sorry. As mentioned, there's a lot of very scary words being thrown around in the media, flesh-eating ulcer. It's been called a flesh-eating Buruli ulcer virus as well. And there's a lot of, I guess, miscommunication. But there is also a bit of a call for vaccination strategies in particular, and other preventative approaches that go beyond just using mosquito repellent. As part of my PhD, I was interested in looking at vaccination approaches. And M-bovis BCG vaccination has been shown to have some efficacy for prevention of Mycobacterium ulcerans infection And those trials were performed in the late 60s and early 70s in Africa, using a relatively less immunogenic strain of the BCG bacteria. But really, in our research capacity, BCG is the gold standard vaccination. Human studies looking at BCG have been relatively suboptimal. They've been done in a very different cohort to what we might be looking at in Australia, where most of our patients are adult patients, middle-aged people, without the complicating comorbidities that people might have in an African context. But also there's been some interesting research looking at BCG, particularly recombinant BCG with immunodominant antigens that might be able to stimulate the immune system or tickle the immune system a little bit better. And if we were going to do a BCG-related randomised controlled trial in Victoria, we'd need to recruit somewhere near 100,000 participants to be able to do that. And thanks to Bridget McNamara for helping me with the statistics here. And something else that you can see in that map is that the Buruli ulcer epidemiology changes very quickly. So if we rolled out an RCT to try to test a vaccination like BCG across one part of Victoria, 2 or 3 years later, it's completely changed epidemiology and it's across the bay. So we probably wouldn't be able to do that. It would be a very expensive clinical trial. It would take many years to actually perform and may not get any feasible result in the end. We've actually got a fantastic opportunity here at the University of Melbourne and as part of the Doherty Institute, a clinical trials unit that has been established and set up for predominantly the purposes of human challenge trials. Human challenge trials are not a new thing. They've been around for decades and they've been implemented for a number of different pathogenic organisms, including typhoid, falciparum malaria, a range, dengue, a range of tropical diseases. They're probably particularly useful for more neglected tropical diseases that are relatively rare or uncommon or under-resourced. And in fact, for us to be able to test something like BCG, rather than rolling it out in an epidemiological study or randomised trial, we could actually test it in as few as 28 people and get that result within 12 months, which is probably a fantastic time frame considering how quickly cases are continuing to go up. So I think we need to be ambitious, we need to be bold, and we need to think outside the box. So for those who've never heard of a controlled human infection model before, we really need to understand the disease better. One of the caveats and limitations of research to date is that most of it's been done retrospectively, looking at patients who've already got Buruli ulcer. No one's really looked or been able to look at prospective patients, taking immune samples, taking clinical histories and looking at that very early disease process, which is probably the most important for vaccination approaches or other preventative strategies in general. And they're particularly valuable for complex diseases because they can be quite difficult to understand without detailed investigations. Not only can we profile the immune system early on, but we can actually use that information to look for underlying correlates of disease or correlates of protection so that we can then go back and better inform our research and development, particularly as it relates to vaccinations. One thing that we've done in the last couple of years is identify a suitable challenge agent. So we've got to make sure that we do this in a safe and a controlled way that's treatable and curable, and that patients or participants aren't left with any long-term consequences of their challenge. So this particular agent that was mentioned yesterday by Kim Blasdell is called M. ulcerans JKD 8049, isolated here in Victoria in 2004. This agent we've published on because we've worked it up and characterised it in great detail to show that it is a suitable challenge agent. Firstly, it's susceptible to the commonly used antibiotics that we would use in the clinic, rifampicin, clarithromycin and fluoroquinolones. We're able to culture it in a minimally toxic medium that doesn't contain tween or other surfactants. We've been able to declump it, which is one of the great challenges, which my colleagues know all about. We can create a large cell bank where each one of those cell bank vials has approximately or statistically the same number of bacteria in each vial. We can genomically show that there's very little genomic change over time or no genomic change over time in its profile, and that we have demonstrated that it's able to actually cause an infection in human macrophages in vivo. One of the things I want to prove is that we can actually take this sample and infect mice. And just as a kind of an example, most of the research performed to date has used incredibly high challenge doses. I was talking to Rachel about this yesterday. We're talking 10 to the 5, 10 to the 6, sometimes 10 to the 7 CFU per challenge. What I'm really interested in is looking at a more realistic natural model of infection that reproduces what we would expect to see in the environment. And Tim Stenier and others before me had done some fantastic work trying to show that very low challenge doses are required to actually create an infection. But what I was really interested in is actually creating those cell bank vials with a very low number of bacteria. We're talking about 10 to 20 CFU per challenge rather than that 10,000 or up to 1 million CFU. So what I really was able to show was that I could actually create a methodology, create a methodology to actually infect mice. This is unpublished work, but looking at the immune response to a very low dose challenge as opposed to the high doses that we've done before, and infecting mice subcutaneously, which is similar to what we'd like to do in a human challenge model. And this is just work to show that BCG does have some protective effect against the incubation period in BALB/C mice, but it doesn't actually change the clinical course of the disease once it sets in. So, but that's relevant because this is the strain that we want to use in humans, and we're interested in BCG as a preventative strategy. One thing that I did do as well is look at the difference between using 100 CFU per mouse and down to 10 CFU per mouse, looking at both B6 mice as well as BALB/C mice, because they've got a very different immunological profile in terms of T helper 1 and T helper 2 responses. And just as a proof of concept, what this shows is that you've got adaptive responses on the left and innate responses on the right. And you can actually see in the splenic tissue, as well as draining lymph nodes, the low-dose model, as you can see, each one of those rows is a different immunological feature, a different adaptive immunological feature, and this is normalised between 0 and 1. But what it really tries to show is that the low-dose model actually captures an interesting immunological profile where splenic tissues are somewhat there's higher cell numbers in the spleen earlier on in the course of their illness in comparison to later on, where the high dose doesn't actually demonstrate that. So you don't really have that resolution to be able to detect a difference in the immunological profile, which is one of the reasons why I really want to use a low dose challenge model to show that we can detect some immunological difference. So I'll wrap up in a minute, but really looking at what an overview of the actual model will look like, and this has been published very recently, is we are thinking about recruiting 3 first-in-human challenge participants and inoculating them in the forearm because it's probably the easiest part to excise, surgically speaking, and then once you've excised it, you just close it primarily. Once 2 out of 3 participants are successfully infected, we'd like to progress on and do a dose confirmation trial where we actually challenge 10 participants to confirm the actual dose. So starting off with a very low dose of 10 to 20 CFU and then progressing as a part of our dose escalation. We're really looking at the primary outcome of safety, tolerability and cure, as well as establishing infection in at least 60% of our human challenge participants. And then we're going to surgically excise all of the lesions and then give them a very short duration of antibiotics just to mop up anything that might be there as a residual. And this is just to summarise that the protocol's available online. There's a lot of information there for you to read about eligibility criteria. Really selecting participants that are at low risk of severe disease is a priority for us. So that's one of the things you can read in that paper. I also performed some qualitative focus group research, and one of the key themes that participants or potentially eligible participants were really interested in is the transparency in communication and ensuring that people are aware of what they're getting themselves into. And this is also work that's hopefully going to be published soon. But I'll stop there because we're probably running out of time. We've got to go to morning tea. One thing that I really love to do is collaborate with others in the audience because, for example, I'm really interested in now coagulation and adding that to the protocol. I'm also interested in looking at mycolactone levels in serum over time. There's a lot of immunological features that we'd really like to understand earlier on so that we can try to target our preventative strategies better. Very open to questions, but we'll probably have to move them to the morning tea.
For questions, but we do have a nice long tea that's coming up, and Tim was going to announce.
We could take. I think because we have
one more
break, we could take.
Okay, we can definitely do that.
Paul Flood.
Questions for Steve?
I'm happy to take all the questions. Yep. Oh, Eugene.
Thanks, Steve. Looks very exciting. I'm looking forward to some of this early work. How will you be using BCG in some of these early studies? Can you explain that? What are you looking for in the subjects?
Yeah, good question. I guess we're just looking to see whether there's any prevention of any clinical disease primarily. We won't have a control group to compare it to, but we'll have, I guess, initially the first 3 participants followed by the 10 dose confirmation participants won't be receiving a BCG. That's just to prove that we can successfully challenge them and that it's safe, it's acceptable. But then hopefully with the BCG we can prove that either they don't develop lesions whatsoever, or maybe that the lesions will actually delay the incubation period, which is the time from challenge to clinical disease, which is what we saw in the mouse models in both the B6s and the BALB-Cs. Yeah. Caroline.
Fantastic work. Thank you very much. I was wondering, when you will be to the stage of selecting the patients, the individuals for enrollment, will you do some assessment of their reactivity to mycobacteria, and to test are they vaccinated with BCG, or will you assess that?
Yes, so we'll be excluding, for the initial part at least, maybe we can change things down the track, but we'll be excluding people who've got a history of BCG vaccination or a scar that could look consistent with BCG vaccination. So they'll be excluded specifically, just in case it confounds the results. In Australia, BCG hasn't been used since the mid-1980s as part of its routine schedule, so given that we'll be only recruiting young adults, people under the age of 45, it's unlikely that they're going to have exposure to BCG. And then in terms of whether we're going to exclude people, we're not testing people serologically for any marker of Buruli ulcer disease in the past, but we will exclude people who've got a clinical history of Buruli ulcer disease, or anyone that lives within 2 kilometres of an endemic area or has lived with other people with Buruli ulcer. So we're trying to sort of using more epidemiological rationale rather than serological. Yep, thanks. Paul. Oh, Rachel. Yeah.
Yeah, thanks, Steve. I think it's really exciting work. In a similar vein to Caroline, I was wondering, there's some SNPs which predispose or protect people from Buruli ulcer in an African setting anyway. Will you be thinking about, well, at least taking DNA so you know what the SNP status is for those
for the participants?
Host defence?
Yeah.
I mean, there were sort of autophagy pathways and macrophage host defence kind of pathways on the whole.
It's not something we've proposed in the protocol, but it's a really good suggestion, and it's something I think we'll have to take on board. I think it's an extra couple of barriers in terms of getting, ethics and getting human genomic material, but I think that at the very least we can think about how we can approach that, but it certainly sounds like it'll be an important...
Yeah, I suppose I was just thinking, how are you going to manage it if someone's injection site progresses much more quickly than you expect, for instance?
Yep.
How do you manage that?
I mean, there's always the small possibility that an inflammatory edematous sort of cellulitic reaction will take place, and so in the protocol we've got, caveat that we've got a definition for how many centimetres that can progress before we call it that, and then if it develops that quickly, we'll straight away be giving them antibiotics to essentially abort the trial. But hopefully our participant selection will also help with that, because we know certain people at a high risk for developing edematous disease, people of older age, young children, so they're excluded, people with diabetes, and also the site of the challenge, so around that part of the body it's less likely, but if it was on the hand or the foot or something like that, it might be more likely. So we're kind of trying to use our key participant selection criteria to minimise that possibility, but you're right, there's always that possibility, yeah.
It's just a comment, but it is hard, isn't it, to make sure no one's exposed, because you only have to enter an endemic area for 4 hours to get, that's the minimum I've seen. But anyway, it's a reasonable approach.
Yeah, thanks. Very happy to chat in the morning tea break. Yep.
All right, could you just join me in thanking our speakers and our chairs for that session? Kingsley reminded me of a strategic error yesterday, that we should have taken the group photo when we had our political leaders here, and that was a bad miss, so I own that. But we should still have a family photo now. So what I would like us to do is, Ange is our photographer. Is that right, Ange? You're going to do this? Yeah, I think our plan is we'll put a generic IMUC background here. Everyone comes up sort of forward here, and Ange will be somewhere there and take our picture. So, and then we go to morning tea. So if we could just all come up the front. You can't all hide up the back as well, so tall people at the back, the usual drill.
It's going to be complicated. People are covered by oh well. Yeah. We're going to start the shuffle. Yeah, but we're yeah. It's okay, I don't know what to do with my hands anyway.
This is wrong. Fearless leadership.
Where to? More people in these tall guys.
Yeah, it's right here.
We need to take a step back. There's room here. We've got room here.
Yeah, there's
Take a selfie.
yeah, it should be.
How's that look, Ange? Yeah, exactly, Rachel.
All right, we might make a start, so let's wrap up. Let's wrap up our morning tea. All right, thanks everyone. Please take a seat. All right, so we've got some fantastic talks coming up. Next up, we've got Mr. Jonathan Kofi Ajayi from Kumasi Centre for Collaborative Research in Tropical Medicine. So Jonathan is a PhD student affiliated with the Kumasi Centre for Collaborative Research in Tropical Medicine at the Kwame Nkrumah University of Science and Technology in Kumasi, Ghana. His research seeks to identify biomarkers with diagnostic and prognostic capabilities in Buruli ulcer disease, with the hope that this would lead to the development of reliable immune-based screening or diagnostic tests, and tests that can work for treatment monitoring in Buruli ulcer. So please welcome Jonathan to the stage.
Good morning, everyone. Thank you to the organizers for inviting me here. And moving away from the very interesting presentations this morning about mycolactone and its immunosuppressive effects, and SEC61, and Steve, your work on the immune challenge model, the human challenge model, we come to some clinical work from KCCR. So as mentioned, my name is Jonathan Kofi Ajayi, and I would like to present my work from KCCR that indicates that patients with Buruli ulcer show a distinctive B cell profile. M. ulcerans is predominantly an extracellular organism because mycolactone's immunosuppressive effect, it's able to break away from phagocytes that try to engulf it. This means that the humoral immune system is indispensable in the control of Mycobacterium ulcerans. B cells, which are critical for the humoral immune response, creates B cells, which are key to the humoral immune response, are very critical for the antigen presentation, for antibody production, and for cytokine production in the control of M. ulcerans. But a lot of work has only focused on T cells. The B cell usually is left in the background. So what we sought to do is to focus our attention on B cells. Historically, cross-reactivity has caused some challenges in the identification of immune cell biomarkers that could help in the diagnosis of Buruli ulcer. So what we hypothesized was that B cells the proportions of B cells in peripheral blood are changed during BU infection, and this could serve as a biomarker for disease diagnosis and monitoring in Buruli ulcer. This has changed. So what we did was to recruit healthy controls and BU patients, and to cater for the challenge of cross-reactivity, we recruited TB patients. This is the characteristics of the patients and healthy controls that we took. We made sure that the characteristics of the groups were comparable based on age and sex matching. For the Buruli ulcer patients, we took 2D wound lesion measurements of the Buruli ulcer lesions. Blood samples were taken from these participants, and flow cytometry that targeted B cells and B cell subsets was done. This is our gating strategy, looking out for total B cells and B cell subsets. The subsets that we were interested in were memory B cells, transitional B cells, naive B cells, plasmablasts, and regulatory B cells. What our results showed was that total B cells were no different in the Buruli ulcer patients compared to the two sets of controls. That's healthy controls and the second set of controls from TB patients. However, when we looked at the different subsets, the 5 different subsets, what we noticed was that regulatory B cells and memory B cells were different in the Buruli ulcer patients compared to the two sets of controls. Whilst regulatory B cells were diminished in the Buruli ulcer patients, memory B cells were elevated in the Buruli ulcer patients. In order to see whether these changes were reactive towards treatment, we looked out for changes during the course of treatment, comparing 3 other time points, week 8, week 16, and week 32. Here again, what we saw was that regulatory B cells and memory B cells were different at the 3 other time points compared to the baseline. Whilst regulatory B cells increased during the course of treatment, memory B cells decreased during the course of treatment. What was interesting was that these changes over time were comparable to the healthy controls that we took at baseline. To answer the question whether these changes were associated with healing, we did a time course analysis of the changes in the surface area. And what we saw was that regulatory B cells negatively correlated with the lesion size. That is to say that whilst the lesions resolved with time, regulatory B cells increased in the periphery. On the other hand, memory B cells decreased as the lesions resolve. This forms a sort of balance between activation and regulation that happens in B cells as treatment progresses. We would like to conclude that B cells play a role in the host immune system during Buruli ulcer disease and possibly immunopathology in Buruli ulcer disease, and changes in the B cells can be used as biomarkers for disease diagnosis and treatment monitoring in Buruli ulcer. I would like to thank the Buruli ulcer team at the KCCR and these institutions for supporting my work. Thank you very much.
Questions? Yes.
Testing. There we go.
Thank you very much for a nice presentation. I've just wondered in these young individuals how you standardize the diagnosis for tuberculosis. This is challenging in these young children.
So for the diagnosis of tuberculosis in our work, we used the clinical symptoms that came with the clinical symptoms. Then we brought samples to the lab that we did an immunological assay to confirm a TB diagnosis. Added to that was a suggestive X-ray of confirming tuberculosis infection.
Got another minute for another question. Yes. Do you want to introduce yourself and tell us where you're from?
My name is Michael Mleme. I'm an epidemiologist at the Bowen Southwest Public Health Unit. Mine is a basic question. Why do you think the regulatory B cells behaved differently from the memory B cells?
What we noticed with treatment was that regulatory B cells increased during treatment. We know that regulatory B cells produce interleukin-10, which controls inflammation. And inflammation is needed for wound healing. So as the wound heals, the need for more inflammation is reduced. Therefore, the need for more regulatory cells in the periphery also reduces. So that's why we saw that.
All right, if there's no more questions, we'll go on to the next speaker. All right, thanks very much.
Okay, our next speaker is Dr. Akira Kawashima from Taikyo University, who is a postdoctoral fellow at Faculty of Medical Technology. He's published papers on innate immune responses in Buruli ulcer and leprosy. His research focuses on how the innate immune system maintains health and under what circumstances it promotes disease. In particular, he studies the molecular mechanisms that activate or suppress the immune system and how they promote cell death and immune activation in various infectious diseases.
Actually, the speaker was changed. Good morning, everyone, and for local people here, good day. Might. My name is Koichi Suzuki, and this study was done by former postdoc Akira Kawashima, but today, I will present on behalf about the role of SEC61A1, which is very embarrassing for me, because it was not very nicely covered this morning by two speakers, Rachel and Carolina. But what we have done was that we tried to identify a key molecule which mediates microlactone action to induce cell death, and well, using genome-wide screening. And we found that SEC61, yes, it is. So that's all. So maybe I can stop here, but if you insist, I will continue. In the previous studies, we have shown that microlactone directly induces cell death in Schwann cells in mice, as shown by swelling of nerve fascicles, and vascular changes of myelin sheath, and also the appearance of tunnel-positive apoptotic cells. and the appearance of cleaved caspase-3 in both mice and human Schwann cells in culture. And not only Schwann cells, but extensive necrosis without inflammatory cell infiltration is seen in skin lesions of Buruli ulcer. This is a Japanese case. And in pre-monocytic THP1 cells, synthetic mycolactin A and B induced caspase-3 dependent apoptosis, as evidenced by the appearance of small condensed nuclei with bright fluorescence and the appearance of cleaved caspase-3. And both cell viability and cleaved caspase-3 were inhibited by a caspase inhibitor, ZDV-FMK. So, we aimed to elucidate the mycolactin-induced cell death pathway by screening for host factors. To do this, we employed genome-scale lentivirus CRISPR mutagenesis assay using THP1 cells. And a genome-wide CRISPR knockout or gecko screening strategy has been utilized. Briefly, lentivirus library consisting of more than 120,000 single guide DNA targeting more than 19,000 human genes was transfected in THP1 cells, and cells were selected by puromycin and treated with mycolactin for 1 week. And genomic DNA was purified from mycolactin-survived cells, and original sgRNA sequence was PCR amplified and sequenced using next-generation sequencer to identify responsible genes for mycolactin-induced cell death. As a result, the left panel is a volcano plot where the x-axis shows the number of enriched sgRNA, and the y-axis shows the p-value between control versus mycolactin-treated cells. This is an analysis from model-based analysis of genome-wide CRISPR-Cas9 knockout. And don't ask me for details. But in total, 885 genes were identified as candidates causing mycolactin-induced cell death, and the name of the top 10 genes was with highest p-value listed. To confirm the role of these 10 genes, each sgRNA was transfected into THP1 cells treated with mycolactin, and viability was evaluated. As a result, only cells with Sec61A1 knockout showed significant survival after mycolactin treatment. And I don't need to repeat this, but for those people who missed this morning session, Sec61A1 is the alpha-1 subunit of Sec61, which is a translocation channel or translocon in the ER membrane. And Sec61 transports newly synthesized signal peptides into ER lumen. And also, it is responsible for the efflux of calcium from the ER. It was previously shown that mycolactin binds Sec61A1 and prevents the binding of signal peptides. It also inhibits protein translocation from the ER to the cytoplasm, causing misfolded proteins to accumulate in the ER and leading to ER stress. And of course, ER stress causes apoptosis. And these studies were performed using based on mutation screening and structural analysis in several different cell lines. So our study using genome-wide screening in THP1 cells supports previous studies showing that Sec61A1 is a target of mycolactin. So we have confirmed that number of mycolactin-induced apoptosis as evidenced by caspase-3 and 7 positive cells shown in red. showing red, was significantly reduced in SEC61A1 knockout cells. And Western blot analysis revealed that cleaved caspase-3 seen in control THP1 cells was not detected in SEC61 knockout cells, while knockout had no effect on the actinomycin D-induced caspase-3 cleavage. This evidence again indicates that SEC61A1 is responsible for mycolactin-induced caspase-3-dependent apoptosis in THP1 cells. And it was known that dysfunction of SEC61 leads to accumulation of misfolded proteins, thereby activating ER stress-related genes, which in turn activate pro-apoptotic genes. So we therefore evaluated the mRNA expression of genes related to ER stress after mycolactin treatment in THP1 cells using real-time PCR. As a result, mRNA levels of early response ER stress-related genes and pro-apoptotic genes were significantly elevated in control cells following mycolactin treatment. However, this elevation was completely abolished in SEC61A1 knockout cells. Also, mycolactin-induced phosphorylation of eukaryotic initiation factor EIF2 alpha, which is known to lead to apoptosis. But this phosphorylation was reduced to its original level in SEC61A1 knockout cells. So in summary, we have performed genome-scale lenti-CRISPR mutagenesis assay using THP1 cells, and we cloned SEC61A1 as a responsible factor for mycolactin-induced cell death. We have shown that SEC61A1 mediates the action of mycolactin to activate early response ER stress-related genes and pro-apoptotic genes, activate caspase-3, and phosphorylate the transcription initiation factor EIF2 alpha. And Rachel said this SEC61 will not be a target for therapy, but it will be possible to suggest that inhibition of mycolactin activity or its downstream cascade could be a novel therapeutic modality to eliminate the effect of mycolactin, but I'm not going to say it loudly. Thank you very much.
Thank you for nice presentation. Any questions?
Thank you very much. My question is about the SEC61A1 knockdown mutant that you made. Did you notice, because I think this gene is essential, so in the just without adding mycolactin or anything, did you see a difference in the percentage of viable cells in your culture dish? And was it because you present the graph as relative to control, but in the control conditions, were they comparable in terms of viability?
Yes, in other cell types, it was reported that the SEC61 is essential for cell survival. But our assay strategy is to purified sgRNA sequence from mycolactin survived cells. So in our system, knockout of SEC61A1 reduced cell growth rate, but still survived, not completely died.
Yeah,
maybe it is
for a few days at least. Yeah, for the time, that is sufficient to perform the assay.
Yeah, that could be a different in cell types or different concentration or treating times.
That's it.
Okay, thank you so much.
Thank you.
Okay, next up we've got Dr. Belinda Hall from the University of Surrey. Move your slides forward for you. There you go. Belinda is a Senior Research Fellow at the University of Surrey in the UK with a Bachelor of Science in Biochemistry from the University College Cardiff and a PhD from the University of London. She has worked at EMBL, the London School of Hygiene and Tropical Medicine, Tufts University, Imperial College and Queen Mary's University in London. After initially researching cancer, she moved into parasitology, working on cell biology and host pathogen interactions in malaria and trypanosomiasis. Apart from the odd foray into virology, she has been investigating Buruli ulcer and mycolactone since 2011 and is currently studying interactions between Mycobacterium ulcerans and host macrophages. So please welcome Belinda.
Right, so thank you for the introduction. Thank you for letting me talk today about our work.
Yeah, thanks again to Belinda for that fantastic presentation.
Okay, our next speaker is Mr. Kwabena Awusu-Boateng from University of Surrey, who is currently a PhD student in Rachel Simmons' lab, studying the interactions of mycolactone-producing mycobacteria and macrophages. He was previously a student earning his MPhil in Lydia Mosi's lab. So, Kwabena.
Okay, so now I've got the pleasure of reintroducing my co-host, Professor Heather Jordan from the Mississippi State University. So I am going to introduce you though, because I think you've got some fantastic work to present. But so Heather's a professor in the Department of Biological Sciences at Mississippi State University and co-chair of the WHO Skin NTD Sub-Working Group on Transmission. Her research focuses on field studies to understand the ecology of Mycobacterium ulcerans and other mycolactone-producing mycobacteria in natural aquatic environments and lab studies to measure microbe-microbe interactions between M. ulcerans, mycolactone and other skin pathogens, and host microbe interactions and forensically relevant bacteria, also lab foci. So please welcome Heather to the stage.
All right. Thank you. Thank you so much for the opportunity to talk to you all today about some of the work that we've been doing in the lab with M. ulcerans and Staph aureus co-infection studies. But by way of background, we know that in some patients, despite the initiation of antibiotic treatment, there is delayed wound healing that has been shown for some of these patients, where some reasons may include a restored inflammatory response, as we talked about some yesterday, potentially some lasting negative impacts of mycolactone on host cells, or possibly even polymicrobial interactions that are occurring in Buruli ulcer wounds. And this is a really busy slide, so bear with me. This is my rationale on a slide. We know that some pathogens, such as Staph aureus, have been isolated from Buruli ulcer wounds, which you can see in some of these publications here. But despite this isolation, they are lacking that typical Staph aureus pathology, such as pain, redness, pus. Anybody that's had a staph infection knows they're pretty nasty, pretty gnarly. But in many of these cases, there's a lack of that pathology. And as I said, it's pretty interesting that that is the case, because when you look at this this really complex network, Staph aureus is a pretty prolific pathogen, and this complex network that it uses senses environmental signals and cues to respond to these environmental signals through changes in gene expression, modulation of virulence, with some of these phenotypic traits to allow it to survive in a host. Now, there was a study that was conducted in 2017 that I found quite interesting. This is by Amisa et al. In 2017, where they isolated Staph aureus from 21 Buruli ulcer wounds. These are the patient numbers here. And then they looked at the presence or absence of some virulence genes associated with Staph aureus. So for instance, up across here, which you can barely see, you have alpha, beta, delta, hemolysin genes, and some other virulence genes as well. And by indication of these pluses, you can see that there is the presence of these genes in the Staph aureus isolates. Now then, in this table here, they looked at hemolytic activity in those Staph aureus isolates, and you can see through the HLA, B, and D hemolytic activity, there's missing some of these pluses, showing that despite the fact that these genes are present and intact, there is a lack of hemolytic activity in some of these isolates. So again, this was intriguing to me because I have had a staph infection and it hurt, but it made me think about it more, the impact of emulcerans and mycolactone in these interactions. And up here on the right, we've seen the structure of mycolactone, and it kind of, sort of, maybe similar to some quorum-sensing compounds in other bacteria, where some of these quorum-sensing compounds are known agonists to bacterial competitors. So for instance, here is a study that was showing that Pseudomonas aeruginosa quorum-sensing molecules can lead to inhibition of Staph aureus quorum sensing. So it actually led us to test the hypothesis that perhaps mycolactone is an agonist to Staph aureus virulence, quorum sensing, and possibly even growth. So this work was conducted in my lab by students Lakshmi Dungal, Albert Cujo, and Lindsay Burcham, and it was published in 2021, where they first looked at growth, where they incubated Staph aureus with increasing concentrations of mycolactone, measured OD as an indicator of growth over time. And you can see that with these increasing concentrations of mycolactone, there really isn't that big of an impact on growth, except at 10 micrograms, which is a really high concentration, and we consider probably not biologically relevant. In panel B here, we also looked at hemolytic activity on rabbit red blood cells with Staph aureus that have been incubated with increasing concentrations of mycolactone over time, 3-hour, 6-hour, 24-hour, compared to Staph aureus alone, shown in these hatches, or red blood cells in water. And what you can see is that with the concentrations of mycolactone, these 3 concentrations, incubated with Staph, there is a decrease in hemolytic activity, some of which are significant as compared to the controls. Next, we use a luminescent reporter to measure the impact of mycolactone on the HLA promoter activity compared to an ethanol vehicle control over time. And this is with 300 nanograms of mycolactone here. You can see that this is the vehicle control in triplicate. The Staph incubated with mycolactone led to this significant decrease in HLA promoter activity over time. We also used RT quantitative PCR to measure changes in gene expression of certain virulence genes when, again, incubated, with Staph was incubated with mycolactone at 3 different concentrations and also across 3 different time points here. So focusing in on the 6-hour, you can see at the highest concentration, this 300 nanograms in red, it led to a significant decrease in gene expression in AGRA, SAER, and HLA, which are virulence genes in Staph aureus. At 24-hour, we also showed that the mid dose in blue and the red dose also led to significant decreases in gene expression in the SAER and HLA. Now, we also used RNA-seq, where this again is with the 300 nanogram mycolactone incubation. And without going into so much detail, what we found were significantly different changes in gene expression according to functional category across here, also across time at 3 and 6 and 24-hour, up and down regulation, where the take-home really is that there's a profound modulation of gene expression with regards to Staph aureus, broad metabolic pathways, some in virulence, genetic information processing as well. So all of these were very interesting to us. They were exciting as a potential means for data suggesting potential means for these interspecies interactions and potentially a platform for discovering new mechanisms for interactions within these polymicrobial Buruli ulcer wounds. But we do recognize that this is synthetic mycolactone added to Staph, and so we really were interested more so in those cell-cell interactions. How are the pathogens themselves responding? Also, what's the response overall to the host? So we used a guinea pig model of infection to try to get at some of these questions, where we used 16 anesthetized and shaved, partly haired of guinea pigs, and this is our infection model up here. You can see, based on our IMUC protocol, we have 4 different injection sites. Our top 2 were in all guinea pigs, where we had, these are our sterile PBS internal sites, where we injected with sterile PBS 200 microliters using a 25 gauge syringe. Our bottom sites were our treatment sites where we infected with 200 microliters of either emulcerans alone, staph alone, a combination 1-to-1, or PBS as a control. Oh my Lord, sorry about that. Anyway, so we conducted these. We followed them over time, weighing and also monitoring daily. At that 14-day time point, we sacrificed, where we then collected tissues for culture and histopathology and also RNA analyses. So this work was done by my student, Sydney Boatman, along with collaboration with Dr. Alicia Olivier, who is a vet pathologist, which I am not. So I am just going to read what Dr. Olivier found with her histopathology. So with the Staph aureus alone, she found these scattered lymphocytes and plasma cells with fibroblast proliferation in the deep dermis, extending into the skeletal tissue. And what's shown in panel B is just the enlarged rectangle there. In the co-infection, she found mild chronic inflammation of lymphocytes and plasma cells, locally extensive focus foamy macrophages. Granulomatous inflammation is indicated in the asterisk. And again, this is in panel D, it's the blow up of the rectangle. Now, in the ulcerans alone, it's what I consider, based on our other guinea pig work, to be sort of more this classical Buruli ulcer pathology, where there's extensive areas of coagulative necrosis surrounded by areas of fibrovascular proliferation and mixed lymphocytes around the area as well. So we also sent RNA to collaborators at University of Texas at Galveston Medical Center, who some years ago developed this RT-qPCR array for guinea pigs, measuring different targets of immune-related genes. And so we sent the DNA, and they conducted the array and sent us the data. And so I'm going to point your eyes over here first, where you have Staph aureus alone or emulcerans alone compared to control. And you can see upregulation of these target genes as shown in red. So there's an upregulation of a number of these assayed host genes in response to staph or ulceran single infection compared to controls. Now, if you look at the co-infection, you see that the majority that were once upregulated in the single are now downregulated, with the exception of just a couple of these target genes. So overall, these data show marked differences in pathology in co-infection versus single infection, and also our array is suggesting that there's a widespread dampening of the immune response. Now, I would have stopped there, except we received these data just a couple of weeks ago. It's not really a lot of data, but it's hot off the presses, and it's something that we're really focusing in on in the coming weeks. So we sent off for RNA-seq, and after QA/QC, we received the samples back. We used QA/QC after we received the sequences back, used CLC Workbench to measure statistical differences and transcript abundance of guinea pig transcripts. So what we did first was we looked at differences between the internal sites and the treatment sites. And so that's what's shown here with these Venn diagrams. These are differences, statistically significant differences in guinea pig transcript abundances in the ulcerans, the staph, and the co-infection versus those PBS controls. And so we don't know which genes yet. We're digging into those a little bit more, but it's kind of exciting to us to see that there is a difference in the non-infected areas of the guinea pigs with the infected. We were a little bit nervous after thinking about it of, well, maybe the differences may be due to physiological differences between fat or the biogeography of the injection sites. So we were actually happy to see that the PBS internal and the bottom samples showed no differences. We also have looked across between treatments as well. So we have, again, Venn diagrams, and we're, again, mining these data, but we're seeing differences in the PBS versus ARIAs and the ulcerans alone, and co-infection, where there's an increase in statistically different transcript abundances. There's also differences between the co-infection and the staph alone, the ulcerans and co-infected as well. Some of these are unique and some of these are shared. So we are, again, mining these data with the understanding that this is gene expression, and based on these talks this morning, we obviously understand the impact that mycolactone has on post-translational translocation. So we're also, my student is working on metabolomics and proteomics of these as well to try to understand what's going on there. Quickly, I'm hurrying. We also looked at the microbial response, where I'm going to glaze over and just point out some interesting things that we found. We found 108 differentially expressed ulcerans genes, most upregulated during co-infection. Some of these were PE-PPE genes, a serine threonine protein kinase, a protein translocation subunit, some transcriptional regulators, and a polyketide synthase D gene. And in fact, only one gene was enriched in the ulcerans alone. This was FAA1, which is a long-chain fatty acid-CoA ligase/synthetase. And when we looked through some, what the heck could this mean in mycobacteria, we found that in TB, this could play a crucial role in dormancy, antibiotic resistance, and may be involved in activation of fatty acids imported by TB from the host. And so this deserves a little bit more investigation. In our staph versus co-infection, we only found one difference. That's with CLFA that was downregulated in the co-infection. CLFA encodes an SRase adhesin, which is a major virulence factor responsible for clumping. So lots of words here. Basically, really excited about working through these because these are single samples that are allowing us to answer multiple questions. We still have a lot to do. We're mining those data. We're doing proteomics, metabolomics. We also have microbiome data that we've got to sort through. We have plans for repeating this study. This is a single time point study. We're wanting to incorporate early and late time points as well. And last, I'm done, with acknowledging my co-authors on the first paper, Sydney, who conducted the guinea pig work, my collaborators at the Vets Wellness Project and at UT Galveston, and my funding through NIH COBRI. Thank you. And I know I went over time. I know I did.
I think we've got time for one or two questions. That's fantastic work, and really interested to see that co-infection really changing the immunological response. Yep. Oh, Eugene. And then Lydia.
Thanks, great presentation. Obviously still early days with some of the more complex analyses. We often see in patients with Buruli ulcer that staph is colonising the area rather than causing true infection. It's rare to get sort of full-blown associated cellulitis and abscess formation. So how would you interpret that? Do you think the mycolactone is switching off the pathogenesis, which you've kind of alluded to? And the second question is, what about other virulence factors of staph? Are you going to look at all of them?
Yes, so yes. That is sort of what we're wanting to dig into a little bit deeper, is that mycolactone may be suppressing some of these virulence factors. And yeah, we do have, from the mycolactone staph study, there are other regulators that are differentially expressed, and so it's suggestive that mycolactone is not necessarily inhibiting everything, but it's certainly inhibiting some of those key quorum sensing factors. That's sort of what we're thinking. But yeah, it's a bummer that this was only one time point, and that's why the next steps are what's early infection, what's later, so yeah.
Lydia Mosi.
Lovely presentation, Heather. I have two questions, if I can remember both at the same time. First one is, did you look to see in the lesions after the co-infection whether there were other bacteria that came up during the infection? That's the first one. Second one is, in your model, you're putting both test and control on the same animal. Don't you think it's going to affect the gene expression studies that you're doing?
It's possible, but we also have, that's why we have the test and the control, but we're also looking across. So there is that possibility. But for us, the question was more so, what are the differences in that local infection, to answer your second first, differences in local infection versus the broader, what's going on up here. We were just curious to see how that's impacting other areas of the skin. So yes, it's possible, but that it's sort of, this gets to our other question a little bit more, and we're comparing the PBS across treatments and within. To answer your first question, we swabbed the PBS and the treatments every other day, and we sent those for 16S, and we have those data back. And as soon as we're back, actually next week I'm meeting with my students, because that's another question we have, is how does this exogenous, if you will, microbe impacts the microbiome. Yeah.
We get one very last question and then we'll have to move on. Peter Kelly.
Hi, Peter Kelly, a physician from Melbourne. Thank you for a great talk. Looking through so that some of the posters suggest that maybe anaerobes are important in the microbiome studies. I don't know whether that's something you can explore as well, looking at anaerobes and how they're co-infection. Maybe that might explain why the augmentin is helpful for the Spanish group.
Yeah, I think we could actually, staff is a facultative, anaerobe. But yeah, looking at the more anaerobic, we could do that with, how would you do that? I guess you could swab and put it on a plate and incubate it anaerobically and look to see how that occupies. But yeah, that's an interesting idea. Yeah, thank you. I'll take that into consideration for sure.
All right, please join me thanking Heather again.
Thank you. While I'm up here, it's my pleasure to now introduce, Rachel, who is going to be giving a talk for her student, Aloysia, on the nutritional impacts, right? So take it away, Rachel.
Thanks, Heather. Thank you. Yeah, thank you. So it's unfortunately Aloysius couldn't be with us in Melbourne this week. So it's a real pleasure to present to you some of the data from his PhD thesis that he recently completed. And he was supervised by myself and Dr. Barbara Engel at the University of Surrey, as well as Reggie Annan, who's at KNUST in Kumasi. And I'm going to describe some clinical work that was done, of course, with Richard Phillips's BU cohorts in the hospitals around the Kumasi area in Agogo, Tepa and Dunkwa. And I'm going to I've practiced trying to give Aloysius' slides in 10 minutes and I can't do it. So I'm going to skip over this bit because I think we've had quite a big introduction. I just wanted to say that the idea for this study came out of a conversation between Richard and I and Babs many years ago now, when we were thinking about host susceptibility, what might be host susceptibility factors in Buruli ulcer, taking into account something we talked about before, the fact that lots of people seem to be seropositive, perhaps as latent infection, as Francois described, but not everyone gets disease. And that's linked with the fact that I was quite recently started at the University of Surrey that has this international reputation for nutrition research. But of course, we recognised we needed a Ghanaian nutritionist as well, so Reggie became involved too. And the rationale, if you're not familiar with this model was proposed back in the 1960s of the links between nutrition and the immune system, is summarised in this diagram here. So basically the idea is that if you have a diet that's inadequate in macro or micronutrients, you become malnourished, then your immune function will decrease. And that decreased immune function would make you more susceptible to infection, so you might have an increased incidence of infection or severity. But actually it's important to also be aware that if you had normal nutritional status and you had an infection, this could lead to an impairment in your ability to absorb nutrients, and you might become now malnourished as a result of an infection that you have. So it's quite a complicated relationship between nutrition and the immune system. The other thing I wanted to say is that this study started quite a long time ago, and in the intervening period there's been some studies published in Buruli ulcer that's really relevant to this area. The first is a couple of papers from Dr. Kofi and the team in Côte d'Ivoire with Ria Yotsu, the first of which is cited here, that used a nutritional intervention to support and showed that they had got enhanced wound healing in BU patients. And second, a study by Melanie Foulon in Laura Marsolio's lab, where they did an experimental infection in mice that were fed a ketogenic diet, and they found that those mice were protected from M. ulcerans infection. So the aims of Aloysius's thesis were to investigate a potential role for diet in the pathogenesis of BU, and he had three objectives. Now, the first one I'm not really going to talk about just because of time. So this was a systematic review which he undertook to try and understand what we currently know. If we look across the 20 NTDs more broadly, what is known about the interaction between diet and susceptibility to disease. And this was partly done because the COVID pandemic came right in the middle of Aloysius's thesis and all the clinical work was interrupted. And then on the clinical side, there were two prongs to the study. The first was to carry out nutritional surveys of patients with BU and controls that were from the same endemic communities and matched as closely as possible for age and sex in a case control study. And then also a biomarker study that used archived serum samples from Richard's extensive archive of samples at the KCCR to look at some biomarkers related to nutrition and the immune system. So, as I say, I'm going to skip over the systematic review, but I'm certainly happy to talk about the findings of that later. So, in terms of the nutritional assessment, so the dietary survey involved 40 BU cases and 40 controls. All of those individuals had anthropometric measures taken, as well as socioeconomic data. And they were asked a food frequency questionnaire that included 105 different foods. And this gave us qualitative information about the types of diets eaten by people in BU endemic areas. And we also performed what's called a 24-hour recall study. So this is a more quantitative analysis where people are asked with sort of examples of weights and measures, so plastic food essentially, to get sizes, what they ate in the last 24 hours. And this information can be converted into a weight based on these kind of portion size props, and then entered into a nutrient analysis template that includes that Reggie Annan developed based on common Ghanaian foods, which gives you the nutritional content per gram of a typical Ghanaian diet. And this is done 3 times in order to increase the certainty of the data. And then in terms of the biomarker assessment, as I said, these are archived serum samples where Aloysius performed a cytokine bead array for the immune markers and ELISAs and colorimetric assays for nutritional markers, so vitamins and minerals. So I'm going to skip over this. The take-home message from the systematic review is that there really isn't much known about NTDs and diet, and in fact not much research done in Africa at all. And Ria and I are currently, because of our sort of now shared interest in diet and NTDs, are now working on a scoping review with Aloysius and a student of Ria. So that should be interesting when we get, that should come up perhaps next year, right? So in terms of the diet survey, so here are the principal findings from the 24-hour recall study. So what we've got here is from the mean for the for the 40 cases and controls in the overall cohort, the daily intake of macro and micro nutrients, comparing the levels in the BU cases to the controls. I think the important thing to take away is that overall, the BU patients' intake of some micronutrients was quite inadequate. But in particular, if we compare the cases and controls, the intake of selenium, vitamin B12 and zinc was lower in the cases than the controls. So they're eating foods that contain less of these things, despite living in the same endemic communities. The other take-home message is that these people were eating a typical Ghanaian diet, so a lot of carbohydrate. In terms of the balance of carbohydrate, protein and fat, it was relatively normal, but at the lower end of protein intake for a normal diet. So the changes that we saw really were at the micronutrient level. But the 24-hour recall study also allows us to look at dietary diversity. And the way this works is that we have these 15 food categories, which would be things like fruits, vegetables, white meat, red meat. And if somebody ate one of these foods over the 3 days of the 24 recall, they would get a point. And so the dots that you can see here are for the individuals in the cohort. And you can see that some people in the BU cases and the controls only ate over 3 days 4 different types of food. Right, but there's also a huge range, and some of them were eating 10 or even higher different types of food, and anything sort of above halfway would be considered okay. So the diets of these people are poor, but not absolutely awful. But the important thing to notice here is that, again, there was a statistically significant difference between the BU cases and controls, and this is despite it being a relatively small cohort. So we went on to do the biomarker study, and of course we were particularly interested in the markers where we saw a difference in the intake. And I'm presenting data here for zinc and vitamin C. And in fact, vitamin C, if I just go back here, did reach statistically different levels. And in fact, the BU cases looked like it was trending a little bit higher, which is interesting. But when we looked at serum levels of zinc and vitamin C, these were both lower in both for both zinc and vitamin C. So what's been drawn onto these graphs is the normal range that you'd expect in serum. And you can see that a lot of people have lower than expected levels of zinc and vitamin C in their serum, but this is even lower for the patients than it is for the controls. Perhaps because of the small numbers, especially when we broke it down into the different types of lesion or the category, we didn't see too many associations between these and the type of disease, apart from significantly lower levels, even lower levels of vitamin C in patients with plaques compared to nodules. And I think this would be something to look at more closely in the future. Again, if we looked at did statistical analysis on the proportion of patients and controls who had adequate or reduced levels of vitamin C and zinc, you can see again there were statistically significant differences. I think here, interpreting the serum levels is a bit more complicated because here you might have some effect of other infections on these levels. And one thing that we thought about in particular, and came up during Aloysius's thesis, was the role of enteric dysfunction, which is these kind of persistent helminthic infections, which can particularly affect young children's ability to absorb foods. But in the cytokine analysis that I'm not showing here, we didn't see any differences in IL-6 levels between patients and controls, and we didn't see really high CRP levels, which I think has been found in BU before. There's not this systemic inflammation that might simply explain these reduced levels. So in conclusion, so despite the high burden of NTDs, we really don't know very much about the role of diet. And I think that's because they affect these marginalised communities who don't attract the kind of dietary survey research that is often done for other more common diseases. And it's really important that we think about this effect of systemic inflammation, and so future studies should certainly measure these factors, and we've expanded on that more in Aloysius's paper. So, yeah, there is this high level of nutrient inadequacy in BU endemic communities for several vitamins, as well as zinc. And we've confirmed that zinc and vitamin C levels are lower in BU cases versus controls. So what's the way forward here? This is something that we're hoping to drive forward. I think it's really important that more work is done to understand the nutritional status of people generally in sub-Saharan Africa, and it does seem like providing nutritional support could be a way of enhancing or supporting patients while they go through their BU treatment journey, and that's supported by the Côte d'Ivoire data. And looking at nutritional metabolites in BU and other skin NTDs would be another interesting area to observe. So as I said, this work is a real collaboration between the University of Surrey and researchers in Kumasi at KNUST and KCCR. And I will leave it there, and I will do my best to answer questions. Thank you.
Okay, just maybe 1 or 2 questions. Peter Kelly, you first.
Hi, Peter Kelly from Melbourne. Thank you, that was a very interesting talk. There's been association between low vitamin D levels and like reactivation of TB. I wonder whether anyone looked into vitamin D levels in this group of patients.
We did look at vitamin D. We didn't see any difference.
Yep. Eugene?
Thanks, Rachel. It's really interesting work. Regarding the control group, you said they were from the same areas or districts. What about the socioeconomic status? Were they matched or different? There was no comment about that.
We didn't see any differences between the two groups in that. So generally they were kind of family members of BU cases, right? I think that's quite a common way of doing it. So they weren't household matched, so they weren't sort of within households, but they were in the same. I mean, these people are poor, right? I think that's true to say, but there was no difference between the two groups.
Yeah, so my point is not a question, it's just something to add to Rachel's presentation. I have 2 points, is that, well, in Cote d'Ivoire, we also see the similar trend in malnutrition, the micronutrient deficiency in zinc and vitamin C in Buruli ulcer patients. But like the zinc insufficiency, it's not just particular to Buruli ulcers, it's also like for wound patients for example, diabetic foot ulcer patients or pressure ulcers, there is this zinc deficiency in wound patients in general. So just to point that out. So yes, one of the treatment for diabetic foot ulcer patient is to provide zinc. So it's already kind of the routine practice for some wound conditions. And then my second point is that also these patients with Buruli ulcers are already malnutrition in the beginning, but also when they visit the traditional healers, one of the kind of the suggestion or the treatment, or I don't know, consultation they get is that they should not be eating meat. And these are given by the traditional healers, which is also one of the, when we do the interviews, that's one of the things that comes up. So it's not just about nutrition, but also the practice of these traditional healers contributing to these results. So some additions. Thank you.
All right, well, we're going to not have any time for questions now, so we can take them over lunch. Thanks again, Rachel, for that fantastic summary.
Now, you may recall there was someone that couldn't do their 1 minute rapid fire session yesterday, but we've got Dr. Yvonne Asantua-Quafor here. Can you come up to the stage? So please welcome Yvonne to the stage. You've got around 1 minute to present your poster. So Yvonne is a dedicated medical lab scientist with a doctorate in medical lab science and a Master of Philosophy in Molecular Biology. She serves at Corlebu Teaching Hospital, providing diagnostic services in microchemical pathology, immunology and haematology. Your research interests lie in diagnostics, tissue regeneration, biotherapeutics, with a particular interest in mycobacterial pathogenesis and treatment in Lydia Mosi's lab. She also researches natural antioxidants, their role in mitigating M. ulcerans toxin mycolactone. So please welcome you to the stage, and you've got around a minute.
Thank you for the opportunity. My research work has been focused on investigating the attenuative effect of natural antioxidants on mycolactone-treated macrophages, raw cells. And what we hope to do has been on whether mycolactone as an inducer of reactive oxygen species, i.e. ROS, can be able to, that we know induces ulcerations and also delay wound healing. Is it possible that readily available natural antioxidants like ascorbic acid, gallic acid, as well as green kombucha tea, be able to mop up or scavenge the ROS such that all these deleterious effects can be prevented? And if these antioxidants are able to scavenge the ROS, is it possible that they can translate into cytoprotection? And if this cytoprotection or increase in cell survival rate is even done, is it also possible that these natural antioxidants can be able to enhance gene expression of endogenous antioxidant genes like superoxide dismutase, catalase, and then glutathione peroxidase. If these antioxidants are able to do that, then it means that there could be increase in the rate at which wounds heal and also prevent the ulcerations. So this is what this project is about. If you want in-depth understanding and explanation to it, meet me at the board and you will get to know more.
All right, it's 12 o'clock, so Tim.
Thanks, Steve.
All right, so now, after all of that, we are going to switch gears. We're going to eat lunch as our next plan. And then we're going to get on a bus. So we have to be on the bus by 1 o'clock. So no dilly-dallying, which, if you're not English speaking, is no messing about. We need to be on that bus at 1 o'clock. And the bus is going to depart from, I believe, just outside the venue here, like on Royal Parade. Correct, Christina? Yep. What else do I need to tell you? Che, I'm not sure if Che's here at this point. He's out in the foyer. Che is going to be our sort of go-to person to help us. If you have questions or you want to know where we're going, we'll all be able to answer those questions for you, but Che is going to be one of our helpers. You can see we're leaving at 1 o'clock sharp. We're heading to Rye. Now, this itinerary, I think, is in your program, so you can sort of see where we're going. You can follow on Google Maps, so if you're worried that you're being kidnapped, you can see where we're taking you. But we're going to Rye. We're going to the Rye Pier. We're going to meet 2 of the local government representatives that have been helping with our investigations of transmission down in this area. They're going to talk about their experience as environmental health officers dealing with Buruli ulcer at a local government level. And we'll get out of the bus and we'll walk around and we will try and find possum excreta. We will encourage you to become skilled at finding possum poo, but we'll see how we go there. And then we're going to do some sightseeing as well. And then we have to be at this, you see there, 1700 hours. We have to be at our dinner venue. We've planned like an early-ish dinner. And then we leave at 8:30 from the dinner venue to get back here about 10 p.m. Now, I'll just check, just so we know, we are providing, if you've registered and you're on the bus, we provide the food for the evening, but drinks, I think, are at individual expense. Yep, that's right. So food is covered, but any alcohol drinks, I think, will be at an individual expense. All right. Is there any questions? Yes, Paul? Yeah. So if you're sun sensitive, like Viking skin, then take a hat and take sunscreen because it's going to be about 29, 30 degrees where we're going with a nice strong Australian sun, hole in the ozone layer, lots of UV. So be sun smart if you have sun sensitive skin. Any other local, any crocodiles, Paul, or snakes? No. Yes. All right. So Rachel has sunscreen if anyone needs it. I do too, so thank you.
And just remember, if you're applying mosquito repellent, put your sunscreen on first, then mosquito repellent. And if you're reapplying sunscreen, then mosquito repellent.
All right. Well, thank you. So let's go and eat lunch. Lunch is out here now, and then we're on the bus. No food or drink on the bus. So do all your eating before. Thanks.